For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 3, Episode 48, our review of some exciting recent clinical trial press releases, starts now.
This week on Surfing the NASH Tsunami.
I don't know the last time ever where you've had this much data report out within a small, narrow window of time and have this much data be positive. Two of the presentations we'll be talking about today are late breaker orals at AASLD. A third one is a late breaker poster at AASLD. And the other one came out before or after the submission deadline, so it didn't have a chance to be presented at AASLD.
Roger, maybe when you announced this episode, call it the WOW episode. It's really good 2 weeks for NASH. Those are amazing results.
Yeah, I'm with Mazen. This is a wow effect for me. This is like the bases are loaded for the phase 3 and you just have to hit the ball out of the park.
Let me throw something controversial, which is the following. We talked about the combo of NITs to diagnose the disease and monitor it and combo for treatments. But the question is, with such a result that Stephen just talked about, do we need a combo or not?
There's combination therapy, and then there's the idea of induction therapy. Hitting them hard, kind of bringing them back from the edge, and then switching them to something that's more palatable for the long term that's oral. That's where I'm putting drugs like this.
You can't underestimate the ability for it to be delivered in an oral mechanism. There are lots of people who just aren't going to want or to use injection. But what's exciting with all of these drugs is all of the different mechanisms. It gives us a great set to be moving forward for, and it is exciting.
My final comment and thinking here is, as Mazen highlighted, liver disease is not all about weight, and I support the concept of having the stool agonism here with some weight loss and some liver-specific effects. This is looking fine.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researchers and key opinion leaders Professor Jörn Schattenberg and Dr. Stephen Harrison, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and today's guest, hepatology researcher and key opinion leader Dr. Mazen Noureddin, as they discuss recent press releases describing the exciting results of Phase 2 clinical trials, this week on Surfing the NASH Tsunami.
So Jörn tells me that this is a big day in Germany. It's the day on which the Germans celebrate reunification, although it's not actually the day of reunification. Do I have that right?
Yeah, that's about right, Roger. And it's a public holiday here, 3rd of October, German reunification. Thanks for highlighting it.
Well, if you're good enough to do the podcast, the least we should do is highlight the holiday. And then to note, and something will be near and dear to Stephen and Mazen's heart, that Jörn was able to do the podcast because he does not have a barbecue this evening.
Yeah, it's getting cold. We had an early dinner. I'm all fed up and set for the podcast here.
Excellent. And so, Stephen, how are you today?
I'm doing great.
Always fun to have you with us, Mazen. Welcome back.
Thank you, sir. Good to be here.
And I came to Louise last because we can commiserate on both our English football teams doing disappointingly this weekend.
We can, but it's nice to see all the boys together.
Yes, it is. It's a big party, right? All we would have to do is get Donna, and it would be the entire band, including all of the leading side musicians. It would be pretty fantastic. So we have a lot to talk about today. When we started the idea for this episode, it was because Akero had put out a really fantastic press release. And by the time we could get it on calendar for this group to get together, Akero had been joined by Poxel, Altimmune, and then after we got it on calendar, Axcella, and then Intercept announced on Friday that they were not filing for cirrhosis because the reverse trial failed to separate. And when you look at the results, it failed to separate. It really failed to separate. So what the 4 trials that did well have in common is the same principal investigator, Stephen, who we're happy to have with us today. So after a groundbreaker, I'm going to ask Stephen to walk us through one at a time, and we'll share what we think all these trials mean individually, ask questions, and then collectively get a sense of what we think it means for the disease and the progression. So with that, why don't we just go to groundbreakers? Brave one, go first.
Yeah, let me start. I mentioned we had the holiday here today, so I'm off clinics. My wife at home. We took for a little family adventure here, visited a castle. We have plenty of them along the Rhine, so whoever is close and is coming to visit me, I'll invite you and take them. It's beautiful, these places, and so I'm all relaxed this evening and looking forward to the discussion after a family day and a day of reunification, which means a lot for Germany, and I think means a lot at this time in particular.
Agreed on that.
Fantastic. Oh, I'll jump in. We're getting ready, obviously, it's Liver Cancer Awareness Month. So we are launching an app called MyLife365.me as of Monday. It should be out. And we're following a Stephen Harrison principle of keeping it simple, in the fact that what we're trying to do is get people to address their lifestyle change and just measure it daily in a really 3 simple characteristics. But as part of that app, you can purchase and subscribe to FibroScans. So as far as I'm aware, it is the first app that allows you to get a FibroScan. And if you are doing any other health mechanism, you can add a FibroScan now to your Noon, your any option if you choose to. Now, certainly in the UK is where we're starting first with that. So with 40 pop-up clinics around the country, we should be able to get earlier recognition and lifestyle management out for liver disease, and even into the medical side. If we're in a location where it's not available, this is what we hope to be driving, a new process to help liver diagnosis or identification, but more lifestyle. So it's exciting for us. It's hard work.
Louise, the we is Towers and Health, right?
We are providing all of the FibroScans, but it's us as Towers and Health app is MyLife365.me, because it's all about my health. It's all about me.
And I suspect at some point y'all are going to hear more about that. Stephen, since you're off mute, why don't you jump in?
Yeah, it's been a couple weeks since I've been here, so lots of things happening. I would say professionally, all kinds of things, as we'll talk about today with these 5 readouts that have come, 3 phase 2B trials, 1 phase 2A, and then, of course, the very large phase 3 well-compensated cirrhotic trial. So it's a great day to be in the field of NASH. Great to be here to talk to you guys today about it, but doing well overall and just kind of in the grind leading up to AASLD, which is just about a month away. So lots going on.
Excellent. Mazen?
Not much exciting. Life goes on. I just had a sick wife and kids this weekend, and I think they're out of their viral infection. And the good news is I'm not infected. So that's my biggest news. But as Stephen mentioned, we're wrapping up for AASLD. We thought we will have a break before October, but we did not. I mean, the multiple projects, multiple papers coming out, and it's going to get busier with the presentations for the AASLD. So a lot of good things happening, keeping you really busy, but at the same time, it's exciting.
That's excellent, everybody. So I guess my good news is, first of all, I said this at the end of last week's podcast, but I'll say at the beginning of this one, sometime in the next 10 days, this podcast is going to hit 100,000 downloads on Buzzsprout. We will throw a party. The party will not be next week, but more about that next week. At least a party online, if not a real party, but we might actually invite people to bring their own champagne. And then second, we're going to show up at AASLD, the podcast, with at least 1, maybe 2 new sponsors who've never sponsored episodes before. 1 committed today. We're working on a second one right now. I'm not going to announce who they are until we've signed, but that's going to happen. That's all exciting stuff. Personally, life goes on. It's a beautiful time of year to be in New Hope, Pennsylvania. So with that, let me turn the floor over to Stephen. And I guess chronological makes as much sense as anything else, but in whatever order you would like to walk us through these trials, let's walk through one, stop for questions, and then walk through the next one.
Thank you, Roger. I mean, again, I think probably important to start off with just some high-level thoughts. First of all, I don't know the last time ever where you've had this much data report out within a small, narrow window of time and have this much data be positive. How positive is it? Well, just to reflect on that a moment, today is October 3rd, 2022. 2 of the presentations we'll be talking about today are late breaker orals at AASLD. A third one is a late breaker poster at AASLD. And the other one came out before or after the submission deadline, so it didn't have a chance to be presented at AASLD. And then maybe what is still out there, the 800-pound gorilla, if you will, is we haven't heard the Madrigal phase 3 data yet. So it's almost like all this groundswell of momentum is building as we ended Q3 and enter into Q4 with the promise of Maestro NASH data being released in Q4. I think it sets us up very nicely for what I mentioned at the beginning of the year, and that is the most exciting year for NASH probably in the past decade. It's a great time to be here and to be talking about this today. So let me do this. Let me go in the order of significance relative to the phases of drug development. We have 1 phase 2A, 3 phase 2Bs, and 1 phase 3. So let me start not necessarily in alphabetical order or in the order that they came out, but with Altimmune. So Altimmune had their top line results were presented on the 14th of September of this year. And this was a 12-week phase 1B study study of pemvidutide. What is pemvidutide? Pemvidutide is a balanced GLP-1 glucagon dual receptor agonist. And so the results from today reflect a 12-week phase 1b study in overweight and obese subjects with non-alcoholic fatty liver disease. So what is pemvidutide? Again, we talked about this being a specific GLP-1 glucagon receptor agonist. And so it does all kinds of interesting things to the body. Through the GLP-1, it induces satiety, it lowers food intake, it also slows gastric emptying. Its glucagon activity works by increasing triglyceride lipolysis, fatty acid oxidation, ketogenesis, downregulating hepatic de novo lipogenesis, and on and on and on, increasing energy expenditure. Somebody put it like this: it's like taking a drug that both induces weight loss and increases exercise without actually having to diet or get out and exercise. So just think of this drug physiologically working in that particular manner. So this, just a study design, I think is important to go over. 12 weeks, 94 patients were randomized 1-to-1 to 1-to-1 to one of 4 treatment arms: placebo, or 1.2 milligram weekly for 12 weeks, 1.8 milligram weekly for 12 weeks, and then a 4-week titration period up to the largest dose, which is 2.4 milligrams. So 4-week titration period, 8 weeks at 2.4 milligram. Important to note, there was no calorie restriction or lifestyle intervention recommended. These patients had to have liver fat content on MRI of at least 10%. And this is in contrast to the other studies we're going to talk about. Because it was an early phase trial, safety and tolerability were key for this in this patient population. So we did not want to have significant fibrosis. So we set a cut point of 10 kPa on FibroScan. Had to be less than 10 kPa, and also had to have an ALT level less than or equal to 75 units per liter. So a bit different than a lot of our other trials. And then they could be a non-diabetic or a diabetic. Classic stuff, had to be on a stable dose of SGLT2 inhibitor therapy, metformin, that sort of thing, for 3 months. And obviously they couldn't be on a GLP-1 because we're giving them a GLP-1 here. So primary endpoint was reduction in liver fat content. Key secondary endpoint was weight loss. Looked at safety, that sort of thing. Just jumping forward to study disposition, 94 patients randomized. There was about 20 patients in each treatment arm. Primary endpoint, reduction in liver fat content at week 12, was very significant for drug versus placebo. Had its peak effect at the 1.8 milligram dose with an absolute liver fat content reduction of 14.7% versus 0.2 for placebo. When you look at that from a relative perspective, that 1.8 milligram dose, 68.5% relative reduction versus 4.4 for placebo. Now, people don't often relate to that. What they relate to is how many hit that magic 30% relative liver fat content reduction that we hear so much about. Ranged from 65 to 94%, depending on which dosing arm you had. The best efficacy was in that 1.8 milligram arm versus 4.2 for placebo. More stringent, 50% reduction. Around 70% of the 2 higher doses hit that versus 0 for placebo. And maybe the most stringent of all, normalization of liver fat content occurred in 50 to 55% of the 2 higher doses versus 0 for placebo. So there were a dose-response relationship relative to ALT reductions in those that had baseline values greater than 30. Weight loss was variable, but when we look at all subjects, it was around 3.4 to 4.3% in this 12-week time period versus 0.2 for placebo. Serum lipids moved the way we would expect them to. Cholesterol was down. Triglycerides were down. When you lose weight acutely, you tend to drop HDL. That's happened here, although not by a significant amount. And LDL was relatively unchanged. Safety, when we look at safety, it's what you would expect from an AE perspective, mainly mild to moderate nausea, some emesis present, although mild. In general, there was also some mild constipation and diarrhea. So these were all GI related and known to this type of mechanism. There were no serious adverse events and 2 cases of AEs leading to treatment discontinuation, or about 4% in the 2 higher dose groups. Importantly, no elevations in ALT that were significant. Blood pressure, heart rate changes were insignificant. And when we looked at glycemic variables and compared non-diabetics to diabetics, no real change here across any of those variables. So in summary, robust liver fat content reductions, significant reductions in ALT, all this occurring at 12 weeks. Weight loss was present as well in both diabetics and non-diabetics. There were no serious or severe adverse events and low rates of AEs that tended to be GI related, and no ALT elevation. So let me stop there and see what the panel has to say about this particular study and mechanism.
I'm going to try to say it once, Stephen. Pemvidutide. I agree. It's a tongue breaker. For me, it stands out that it follows along the metabolic class of drugs with strong safety data here, as you said, and some early efficacy data. The very interesting aspect to me is that it extends the spectrum of GLP-1s by hitting the liver directly with a glucagon agonistic. And I think this is something that is really, from a hepatologist perspective, really key to this MOA for me. And as such, I'm very much looking forward to see that type of class being developed. And I think there is even a broader pipeline on this type of drugs. And I'm going to be excited to hear what Mazen thinks about this, but I think that's reassuring.
This is what exactly in my mind, Jörn. If I caught it correctly, the weight loss was up to 4.3%, which I don't want to downplay that at all. Within 2 weeks, it's very significant, but the amount of liver fat reduction, I think it's slightly disproportional to the weight loss in a good way. It's way more than I would expect from that weight loss. To me, points indirectly to probably direct effect on the liver. So that's the nice thing about it. I think we still have to see in the future the effect on fibrosis with GLP-1 glucagon and GLP-1s. I'm almost certain it's going to probably follow the fat and steatohepatitis, because those are the driver of the disease, and the Kleiner paper and the JAMA Open Network suggested that when you drop the steatohepatitis, the fibrosis will follow at one point or another. Here you're addressing the entire disease as a whole body, not just liver, because you're losing weight and you're improving metabolic parameters. I was curious if what is the rate of cure, and I will define that not just less than 5%, maybe with normalization of liver enzymes. I think that will be a good thing to look at, which will give you a great confidence that the patients are returning to a pre-liver disease status, which is huge in 12 weeks only.
So for those of us who are a little less knowledgeable, how would you normalize liver fat in 56% of patients and not have the enzymes follow at some point? What's the mechanism by which that might occur? I mean, it kind of felt to me like that's night following day. If it isn't night following day, then how would it occur that one would happen and then not the other?
Well, I mean, it's timing is always important, right? So you could, at 12 weeks, you stopped, you're looking at the fat, and there's still a little bit of inflammation that is going to follow a resolution of the fat. So that's one factor. We have seen some data showing a little bit bump in ALT when you defatten the liver, especially quickly. That tends to come down eventually. So that's another factor that will lead to affecting the ALT normalization. So I'm almost certain that with these 55% people, not everyone had ALT reduction, but eventually I think they will go down.
You would guess that, wouldn't you? Louise, go ahead. Or Stephen, do you have a comment first?
Yeah, I agree with Mazen relative to the ALT change. I think it's just a matter of time for that to catch up. I think it's important to put the liver fat content reductions in context here. And you see 14.7% absolute reduction of liver fat content in 12 weeks' time. When we talk about Akero, that's the liver fat content reduction they saw at 24 weeks. And you can see what histology change is linked to that degree of liver fat content reduction from Akero. Different mechanism, but some similar ways in which they work, particularly through the Fgf-2 and 3 pathway, where you're getting upregulation of fatty acid oxidation, etc. So I was surprised that the stock took such a hit after reporting this data, and I suppose that's because Wall Street might have expected a larger weight loss in this population of patients at this 12-week time interval. If you look back at their prior study, there was a little bit more weight loss present, and I think there's a lot of interest riding on this particular drug as an obesity agent. And I think it's important to reflect on a couple things. Number one, if we didn't go through the demographics of the population, but this is almost in contrast to many of the other trials that we'll talk about today. Almost 70% of this population enrolled is Hispanic. And if you look at the liver fat content, again, you had to have a significant amount of liver fat content to be enrolled in the study. And just when we look at the baseline characteristics, again, which I didn't go through, but the baseline liver fat content in these patients were between 20 20 and 24% liver fat content. So when we talk about clinical trials in NASH, that's a large percentage of liver fat content. And that's what you see routinely when you put liver fat content at baseline greater than 10%. You get something in the low 20s. When you set it at 8%, which is some of the trials we'll talk about today, tends to be in the high teens. So when we talk about these normalization of liver fat content in 12 weeks, starting at the level that we're at, that is significant. So let's talk about the weight loss there, because I think that is important. And I think that might be linked to some genetic variables as well, seeing that a significant percentage of these people are Hispanic, probably enriched for P and PLA3 for sure. And the weight loss, it doesn't surprise me that it's only 4% at 12 weeks. If you go back and look at the SEMA data that was published in the New England Journal in their phase 2b trial, and you try to tease out on that graph in the supplemental part of that published paper, and you try to draw a line at 12 weeks, it's around 4% is the weight loss that was seen with SEMA at the same time point. So I think some people are throwing the baby out with the bathwater here relative to this drug. It bodes well that these NITs we're seeing are going to have a significant impact on histopathology when this drug is taken into paired liver biopsy trials. And it's important to note also, I'm unaware of any liver biopsy using a GLP-1 and glucagon agonist to date. Maybe Jörn and Mazen know of something I'm missing, but I'm drawing a blank on where we are with that. So I think this is kind of our first shot on goal, showing non-invasive data, but heretofore, no data with a liver biopsy in that combination yet.
So let me jump in to support that and also remind us to keep on time because we have others. I totally agree with you, Stephen. As a hepatologist, I don't want to see more than 4.3 in 12 weeks. We have this also phenomena that too rapid weight loss might not be good, and it makes you wonder what else you're losing. So another point that if they move that to the cirrhotics, I also don't want to see that rapid weight loss more than what I have seen in 12 weeks in a cirrhotic group for multiple reasons: obesity, sarcopenia, you name it. And we know that. We know that from dieting. Too rapid weight loss is not good in general. So I really like that balance between the weight loss that they achieved in 12 weeks and likely direct mechanistic effect on the liver. And I was just surprised to hear that. This is the first time I hear about their Wall Street effect. I think they should keep going with confidence and their heads up.
My final comment and thinking here is, as Mazen highlighted, liver disease is not all about weight. And I think I support the concept of having the stool agonism here with some weight loss and some liver-specific effects. I think this is looking fine.
Yeah, I was just going to add that is a late-breaker poster at AASLD, so stay tuned. I feel like we're speed dating a bit here. I'm going to jump right into Akero next. Akero is a company that is working in the field of NASH, and they have a compound that is an Fgf-21 agonist. Fgf-21 is an endogenous hormone that has lots of metabolic impact, and they're reporting phase 2b data from the Harmony trial. This is a trial that they're reporting on 113 patients, looking at two different doses, 28 milligram, 50 milligram, given once weekly injection versus placebo. This is a biopsy-proven F2-3 NASH trial, liver fat content 8% or better based on MRI-PDFF. Actually, patients are going to be followed for a year here, or 48 weeks, but there's a biopsy at 24 weeks, and the primary endpoint of this trial is greater than or equal to one-stage fibrosis improvement without worsening of NASH. And there's a whole slew of key secondary endpoints that we probably don't have time to get into today, any more than just a high level. Just jumping right to the chase here, I mentioned how many people were enrolled. About 70% of people were diabetic. About 2/3 of patients were F3. The mean liver fat content was around 17 to 18%, as noted already. It is important to note, and I think any trial that looks at histology that doesn't report on this is probably should be looked at with a jaundiced eye, no pun intended. But I do think we've learned a lot about histology, and we know kind of where some of our pitfalls are. So the way that this analysis was done is that there were two different pathologists. They scored the data independently on an ordinal scale. If they agreed, they reached consensus, that was the final score. If they had different scores, they had to get together and see if they could agree. If they agreed, great. If they didn't, then there was a third pathologist brought in to adjudicate. Now, it's important to note that this did not use AI digital pathology in this particular assessment. So what did the primary endpoint show? Fibrosis improvement of at least one stage with no worsening of NASH at week 24. High dose 41%. 38%, low dose 39%, placebo 20%, treatment effect delta 19 to 21%, significant for both doses. All right. What about NASH resolution and no worsening of fibrosis? One of the key secondary endpoints, 47% low dose, 76% high dose, 15% placebo, significant at both doses. Now, what about the more stringent endpoint of both fibrosis improvement and NASH resolution? 29% low dose, 41% high dose, 5% placebo. One more, patients achieving fibrosis improvement of at least 2 stages and no worsening of fibrosis at 24 weeks. It was 16% low dose, 15% high dose, 5% low dose. So a tripling of that number. Liver fat content reduction, another key secondary endpoint, 6% for placebo, 52% low dose, 64% relative fat content reduction at 24 weeks. When we look at those that had at least 50% liver fat content reduction, it was 63 low dose, 77% high dose. When we look at normalization of liver fat content, 34% low dose, 51% high dose. ALT significantly reduced in a dose response manner. It was rapid and sustained. Pro-C3 using the ELISA assay, not the new Roche Amplicor assay, about a 5 nanogram per ml drop with both doses. ELF scores dropped by more than half a point. Liver stiffness at the high dose, 4 kPa, low dose, 2.6 kPa. Side effects, again, we've come to expect from this Fgf-21 compound, mainly mild to moderate GI-related diarrhea, nausea, increased appetite, again, something we've seen with this mechanism, and increased frequency of bowel movements. And then there was positive impact on HbA1c, about half a point in diabetics, 0.4 if you look at all of them together. Triglycerides were down 25 to 29%, LDL down around 8%, and then HDL up about 24 to 30%. Interestingly here, body weight in the high dose group dropped about 2.9 kilograms. And let me stop there and throw this open for a comment.
Roger, maybe when you announced this episode, call it the wow episode. It's really good 2 weeks for NASH. Those are amazing results. The first study, the second study. Indeed, let me throw something controversial, which is the following. We talked about the combo of NITs to diagnose the disease and monitor it, and combo for treatments. And I don't want to get into that, but the question is, with such a result that Stephen just talked about, do we need a combo or not? This is one of the best results we have seen, given it's a small sample size. It's biopsy proven, really clean, had a lot of things implemented in it that we have been talking about for some time. The 2 pathologists and confirming with the 3rd pathologist, they hit all the primary endpoints that the FDA is asking for, including the NASH resolution and fibrosis improvement. There were 2-stage improvement in fibrosis, and I used to be skeptical about fibrosis improvement in such a short time. In contrast to Stephen, was always optimistic about it, and I think I'm now more optimistic about it after such a result. So fantastic results, very encouraging for those that doubted the field of NASH and the efficacy of drugs. Here we are achieving what we need to achieve with the old, strict, difficult endpoints that has pathology issues, and yet we're moving the needle. So congratulations to Akero and you, Stephen, on this great study.
Louise, you have a comment?
Yeah, just to follow on from Mazen on this one specifically, and probably the previous one, it also gives us data with non-invasive technologies that all correlate potentially with the biopsy-proven outcome. So it could help us push the use of non-invasive technologies with the FDA. It goes back to the conversation at Paris NASH that a lot more data is collected than is used for the production of those endpoints. Where patients sit, moving beyond the biopsy, the more trials that we get, particularly the encouraging data from these two that support that, because all of the non-invasive technologies reduced in somewhat, particularly FibroScan, particularly MRI, PDFF. That for me is encouraging. It means patients and people will engage a little bit better in the trials, and both of them were extremely exciting. I would like to also see CAP data discussed, because of course we're measuring liver fat and we're looking at liver fat by other mechanisms, so I'd just like to see that data included as well. But that's a personal preference, because we are going to need to measure these drugs should they pass all of the rest of the assessments and get out to public use and real world. But very exciting given where we were 18 months ago.
Yeah, and with Mazen, this is a wow effect for me. If you look at the more stringent combined endpoint resolution of steatohepatitis and regression of fibrosis, placebo is 5%, okay? We're not seeing overinflated placebo effects here. And still you have, 30, 40% over placebo in the high treatment arm. You see a clear dose response. I mean, this is like the bases are loaded for the phase 3 and you just have to hit the ball out of the park.
So, Mazen, you said something, you want to say something controversial. Stephen, just one thing and quickly. You're the second one to do that. Stephen, I don't remember if you were on this episode or Louise, but we had, in fact, you both were. Kitty Yale, Chief Development Officer at Akero, was with us for the pre-meet on the FDA call in January of 2021. And in that discussion, part of what came up was the solution was ultimately going to be combination therapy. And Kitty said, we may choose to differ. She was pretty firm about that. And at the time, I said, gee, that's a gutsy thing to do. But looking at these data, you can understand if these data hold up, why, as you say, monotherapy might not be a controversial thing to take a look at.
Yeah, I like to throw controversial things here in the rocket.
I think it's a little premature to say where we're going to use this drug and how it will be used. I do think the results are very positive, but just to be balanced here, there are some concerns with this particular mechanism about tachyphylaxis and, developing anti-drug antibodies. And so I think it is prudent to continue to follow. And I like the trial design here because patients, we're not done, right? We're going to follow these people out for another 6 months. We're not going to do another liver biopsy, but we'll have all the NITs. We'll be able to see if those changes, those positive changes we're seeing are still improving. Have they plateaued? Have they started to worsen again? And we'll have these anti-drug antibodies. We'll have ways of assessing for a plateau effect or even a regression. Good study design, intuitive, thoughtful about where we still have unanswered questions with this mechanism, but positive data nonetheless. So I just wanted to throw that in. There's also a well-compensated cirrhotic trial with this drug underway called the Symmetry trial, and they did comment in the press release that results should be out in the second half of 2023. This is a late breaker, oral at AASLD, if you're interested.
Stephen, real quickly, just one question that you just brought up, which is that if I recall the peg-belfermin results correctly, peg-belfermin did regress. Is there any reason to think, based on the differences in the drugs, that this will have a better outcome? Or is that something we're just going to have to look and see?
Yeah, I think so. They're completely different FGF21s. Not all FGF21s are created equal. This is a fusion protein. It's just wired differently from the beginning. It works on all FGF receptors except 4. That's an FGF19 FXR type mediated pathway. So the 21s don't work on FGF4, but this particular compound has balanced potency against 1, 2, and 3, whereas I believe the BMS was probably weaker on 2 and 3, although don't hold me to that. The other thing I would say about combination therapy, there's combination therapy and then there's the idea of induction therapy, hitting them hard, kind of bringing them back from the edge and then switching them to something that's more palatable for the long term, that's oral. That's where I'm putting drugs like this. The juice is worth the squeeze so far, no doubt. The view is worth the climb. The harder the climb, the better the view. These are injectables. They do have some GI tolerability, although not severe. But we're talking about this is a lifelong disease. So we have positive impacts like this in such a short period of time, and these are sustained in a phase 3 trial. To me, in my clinical practice, I could see using this drug for 6 months, maybe a little longer, having a positive impact, assessing that with some of our NITs that we have at our disposal, and then potentially switching them over to something that's better tolerated for the long haul. And then if they have to go back on it for whatever reason, we could cycle it back on there or whatever. We're learning from the GLP-1 class that people don't tend to want to take these for the long haul in many of our patients. So how do we optimize its utilization to the best benefit? I think that's what we need to learn how to do with this class of drug.
And I would still echo that. We have talked about this antibody issues before in a previous episode. We talked about, could we look at something like the IBD drugs and all that, and that justify the concept that these drugs should not be ongoing for a long time in induction therapy versus switching to something else. Yet, with these encouraging results, you could have people normalizing or getting much better within the induction period, and then they can rely on weight loss and exercise. And I quote the two stages fibrosis data. If someone with F2 gets that, you may argue that some of people that they will normalize quickly. So a lot to see. I think the efficacy, I'm glad we're talking about high efficacy now, and if we need one drug versus 2 drugs again, and talking about side effects and how we can mitigate those and do the least duration. So definitely on a better spot today.
All right, I better keep rolling because we have 13 minutes to go through 3 more programs. Yeah, we'll try to get through on time, keep our audience, at the edge of their seats, which is important. So let's roll over to another paired liver biopsy phase 2b trial. This one with Poxel. Poxel. So Poxel has a trial that's called Destiny 1. What a name for a trial, right? So this is a paired liver biopsy 36-week study, phase 2b trial. To me, it reminds me a lot of the phase 2 Madrigal study. 36-week trial. Liver biopsies were done at baseline and end of treatment. The primary endpoint, however, was PDFF. Now, as opposed to Madrigal, which was PDFF at week 12, this is PDFF change at week 36. Liver biopsies were done, so there's going to be histology here as well. But just to give you a brief intro to the drug, PXL065, that was studied in this particular trial, this is going to introduce a new word for many of you. It's called deuterium. So this is a novel proprietary deuterium stabilized R-pioglitazone. So if you remember pioglitazone, it's a PPAR gamma agonist, been studied extensively for NASH all the way back to, before the hills got dusty and I was a junior fellow that looked much younger than Jörn and Mazen. It's been around for a long, long time. So we know pioglitazone is a 1-to-1 mixture of two mirror image compounds, an R and an S stereoisomer. And using deuterium, you can stabilize each stereoisomer and characterize their different pharmacologic properties. And so in vitro, PXL065 was shown to target essentially the one particular isomer here. So that was the R. R is where you get the biological activity. S tended to be associated more with the side effects we would associate with pioglitazone, weight gain, fluid retention, that sort of thing. This is like taking the best of pioglitazone and joining them together with deuterium. That's why we say it's a deuterium stabilized R-pioglitazone. Knowing that, let's talk about the study. 117 subjects, 3 different doses versus placebo, so 4 cohorts of patients. Primary endpoint, liver fat content reduction at week 36. Key secondary endpoints on histopathology. So just to jump right in on the results, liver fat content reduction at week 36, 21 to 25% reduction. When we look at that magic 30% relative reduction, 40% of the overall treated population hit that number. And that was statistically significant. It did meet its primary endpoint. Key secondary endpoints started with fibrosis here, 31 to 50% in the drug-treated arms versus 17% for placebo. When you look at all of them together, you pool all the treated arms together, 39% had a one-stage improvement in fibrosis versus 17% for placebo. Now, worsening of fibrosis, I think, is an important topic as well. It was 9 to 12% for the treated arms at 36 weeks versus 26% for placebo. At least a 2-point improvement in an NAFLD activity score, 50% for the treated arms, 30% for placebo. Focusing on NASH resolution and fibrosis improvement, that stringent double endpoint, 26% for drug-treated groups versus 13% placebo. And this is pooled data again.
Was that statistically significant, the two of them versus placebo?
No. So this wasn't powered for statistical significance. It did not hit the stat sig button. So more just a numerical trend at this point. One of the important points that people pay attention to with this is if you want to differentiate yourself from pioglitazone, how well do you do that on the AE profile? And I just wanted to mention from a body weight perspective, the press release commented there was no dose-dependent increase in body weight and a minimal least squared mean increase of about 0.68 kilogram in the high dose 22.5 milligram dose. So again, positive results here in this phase 2b trial, particularly on the primary endpoint, but there was trends on key histopathologic endpoints, particularly fibrosis here, that I think deserve some discussion and attention. It turned out to be well tolerated with minimal weight gain to no weight gain. So let's open that up for discussion.
If I think of pioglitazone and talk with Ken Kuzi or people who've been using that for some time, they always stress the concept that this is making your fat tissue more healthy. So I think it's a different mechanism here where the liver is being cured through some effects that we also get off liver tissue. On the other hand, the histology data for the pio has been really supportive in the New England Journal paper, and still some people use it. And so I think this is a refinement here with good tolerability and then potentially maintaining the efficacy, something that'll be very interesting and probably adding to the armamentarium we're seeing that we might be using in the future, because it's compared to the other compounds you discussed, again, a totally different MOA speaks to metabolic, healthiness, if you like.
I mean, I'm along the same line, and Stephen, you brought up great points. It's interesting we presented Akero first and then Poxel, and I just want to point out that this is an oral drug compared to injectable drug, so don't underestimate that. And the efficacy, I think it's excellent of what they have achieved. They got what they needed to get. The side effects are mitigated compared to other PPARs. They have efficacy, and there's a positive signal with an oral drug. So this is definitely a player that needs to continue. And I go back now, and this is what I would like to see in a combination therapy with induction therapy and this drug, or followed by induction therapy. So congratulations to this work, and I think it's very promising. Another positive results.
You can't underestimate the ability for it to be delivered in an oral mechanism. There are lots of people who just aren't going to want or to use injection. But what's exciting with all of these drugs is all of the different mechanisms. It gives us a great set to be moving forward for, and it is exciting for people coming into trials and looking to recruit in the future years. Then there's certainly more excitement and opportunity now.
I want to hark on Louise's point, which is that the more different modes of action you get into the market, the energy and the dynamism around the market will increase exponentially. Because first there'll be a debate about which mode of action matches which patient, then there'll be a debate about how do you put mono and combo therapies together. And it leaves lots of room to play and lots of things to discuss. And given the energy evolving in the class, all these issues, I think, really make for fantastic discussion and a lot of innovation, a lot of investment, and a lot of education.
And I think it's also important we talked about the mechanism of action, we talked about the route of administration, talked about the AE profile, we talked about efficacy. It's also important to point out, and I didn't mention this, but it is there, the change in hemoglobin A1C. That's not insignificant in this population. So that's also when we step back and we take off our myopic glasses as hepatologists, where we're really focused on the liver and we look at the whole patient. Many of these patients are diabetic or on the verge of becoming diabetic, and improving hemoglobin A1C, glycemic control, is important. And so we're seeing early signals that that's happening here as well. So I would be remiss if I say I'd like, the liver fat content reduction we're seeing here is not as good as what we saw with the other two injectable compounds. But again, this is a very heterogeneous disease. Not all patients are created equal, and we need to have an armamentarium of drugs in our toolbox to be able to reach to help manage these patients. And I think if you look at the totality of the data with what we're seeing here, this drug sets itself up very nicely for either combination therapy or even once again, as you induce these people with a more potent drug, switching them to something that's better tolerated for the long term. Maybe this sets itself up very nicely for that as well.
Yeah, Stephen, in that context, I think it's worth noting that before we had GLP-1s and SGLT2s, pioglitazone was a blockbuster successful agent for the treatment of type 2 diabetes. It was called Actos. It was widely used. It peaked at a couple of $1 billion in sales. Go ahead.
Look, I was thinking about this earlier. You go all the way back to the Pivens trial. Jörn was in grade school when that trial came out. That was back in 2010. Long time ago. And what's in the guidance document? Well, you can use vitamin E if you're not a cirrhotic or you're not a diabetic. You know the treatment effect delta with vitamin E in that 2-year trial? The treatment effect delta was 20%. And it's in our guidance. So again, I think we need to understand kind of where these drugs are relative to this very complex and complicated disease. Okay, do we have time for 2 more quick discussions?
Fire away.
We're going to make time.
Yeah, go for it.
We got it.
We're going to make it the WOW episode. But I think Jörn was doing a movie back then.
Yeah, in fact, Mazen, I was going to ask you if that was when he was making Good Will Hunting, but you beat me to it.
Oh, my gosh. All right, guys, let's talk about... wait. I needed to update you on the Poxel. So that is also a late breaker oral at AASLD. So good news for them. Okay, so now moving on to Axcella. So Axcella, I feel like this one is flying under the radar a little bit. And so I'm glad we're going to talk about it. So Axcella has a group of endogenous metabolic modulators that are trying to get at in a pleiotropic manner these multiple different pathways that regulate NASH pathogenesis. And it does that by combining in a proprietary way various amino acids in combination that target some of these pathways. It's an oral drug. It's like you mix it up in a drink. It's like, I don't know the best way to say it. If you remember Tang, the breakfast drink, it's a little bit like Tang. And you take it twice a day. So this trial, it's a phase 2B paired liver biopsy study. About 270 people are going to be enrolled in this one-year trial. It's a very good sized phase 2B study. It's underway. It's currently enrolling, but they had this pre-planned interim analysis, much like Casabutate did in their phase 2B study that I'll read out in the Q1 of 2023. So here we have this interim analysis when 30 subjects in each arm reached 12 weeks. And so that happened. That's what was pressed the other day. There are two different doses of what we call AXA1125, 22.6 twice a day and 33.9 twice a day compared to placebo. For this interim analysis, there was roughly 40 people in each arm. And just in summarizing the patient demographics and baseline characteristics, this study population is reflective of a very active disease state with significant fibrosis. Now, in this interim analysis, all that's being read out are 3 non-invasive markers: MRI-PDFF, ALT, and FibroScan. That's what I'm going to tell you about here. So just jumping to the chase, when we look at FibroScan, they're reported at 12 and 24 weeks because there actually were about half of those patients, maybe a little more, that made it all the way to 24 weeks and were included in this interim analysis. So at 24 weeks, you see a 4 kPa drop in FibroScan with the high dose and 2 kPa drop for the low dose, and actually no change in placebo. This was statistically significant for the high dose. So the 4 versus no change was significant. Now, putting that in perspective with another company we just talked about on this podcast, Akero, at 24 weeks, that's the change they saw in FibroScan with the high dose, 4.3 for efruxifermin 50 milligram. It was 4.07 for the AXA1125. When you reflect on what that is compared to maybe other drugs, the GLP-1 semaglutide at 72 weeks did not show a change in FibroScan. We look at obeticholic acid. We'll talk about that in a minute. The 18-month trial had a 1 kPa difference with the high dose, 25 milligram dose. So just reflecting on this particular biomarker, I would say that's very promising. What about ELF and FIB4? Those were also collected. There was a rise in the placebo of 0.4 at week 24 in the ELF. It was the same in the low and the high dose, minus 0.3, and those were both significant, but compared to placebo in large part because placebo went up. What about FIB4? I'm not a big believer in FIB4 as a monitor for therapeutic efficacy. However, it went up a little bit in placebo, went down a little bit in the drug-treated group in a dose-response manner. It was significant for both doses. And then finally, MRI-PDFF. Let me talk about ALT first. ALT at 12 weeks had I'm going to just mention the change, the group of people that had at least a 17 unit per liter change for the sake of time, because that's the magnitude of effect change that Rohit showed us from the Flint trial. At the high dose, 39% of people had a 17 unit per liter drop. That was not significant relative to placebo, although there was a dose response relationship. Placebo had 17% that hit that magic number versus 39% in the high dose. Again, a trend there, but not significant. What about PDFF? Just looking at a 30% relative liver fat content reduction. That's the magic number we talk about. Around 36% of the low dose hit that number. That is significant relative to placebo, and 29% of the high dose hit that number. I think there's some variability there because of the small numbers of patients at that time point. So let me stop there and open this one up for discussion.
This is another oral, Stephen. As you mentioned, it's a mixture. So it's almost a combo drug. It through its efficacy of multiple amino acids and signaling molecules, it addresses different pathways. I remember discussing it on the podcast, an abstract at ASLD, must be 1 or 2 years ago. And I was impressed with the garden variety of the pathways it addressed. And I called it a garden hosening effect, where you have like a broad activity of the drug. And I think that's something we're seeing here. We're seeing effects on fat. We're seeing effects on the non-invasive markers of inflammation. And as such, It's, again, an addition, potentially a very useful one to the armamentarium as we move ahead into the era of treating NASH with pharmacotherapy.
Yeah, and I would be remiss if I didn't. We just got in a hurry, so I didn't mention the safety and tolerability. So a very safe, very well tolerated drug at this point. Some minimal GI tolerability issues, but seem to be well balanced between groups relative to placebo.
Stephen, simple question. How palatable is it? I'm mindful of the idea that there are drugs that you've mixed in this way that, for example, the cholestyramine resins that are really not easy for people to take, and there are others that are quite easy for people to take. So,
yeah, this is nothing.
And by the way, I've heard both described as like Tang.
Yeah, this is nothing like that. Not everybody's a fan of Tang, but most people can tolerate it. If this drug continues to hold up well, and I think it will have a role to play in the treatment of NASH, and maybe they come out with a couple different options on taste. Maybe they have a couple different flavors or something for people. But
Louise, any question on that before we go on to reversing out of here?
No, I like this drug and I liked what it's saying, and the potential is certainly exciting. It just fills me with joy.
Okay. Go ahead, Stephen, on to reverse.
All right. Just recently, today's October 3rd, on September 30th at 8 o'clock in the morning, we did get a press release from Intercept announcing the reverse phase 3 study of a beta-cholic acid in compensated cirrhosis due to NASH. And unfortunately, it did not meet its primary endpoint. So just a little background, the reverse study was a study looking at patients with well-compensated cirrhosis due to NASH. And there were over 900 patients randomized to this treatment, and ultimately they received once daily oral OCA, 10 milligrams. There was also a group that received 10 milligrams titrated to 25 milligrams after 3 months. So it didn't meet its primary endpoint. I mean, we could go into the numbers, but the placebo response rate was a very generous 10% or something like that, and the drug-treated groups were something around 11%, 12%, so not significant. And then when we look at safety, I think that's maybe a take-home point from this that could be leveraged in the new drug application that they announced they're still planning on filing by the end of the year for the non-cirrhotic population, the F2-3 population, based on the readout from the Regenerate trial. So we have safety in 916 subjects who took at least one dose of drug. And what they comment on is that treatment-emergent adverse events and treatment-emergent serious adverse events and deaths were balanced across all treatment groups in reverse, with the most common side effect being pruritus. There's no surprise there. It's the most common reason for treatment discontinuation. I will note that there is a pre-specified point in the protocol where patients have to stop if they're having pruritus. So potentially that number is a little bit more elevated than what you would see in general practice, although that's a bit controversial. When we look at gallbladder-related events, they were balanced across the arms, consistent with the known mechanism of this class of drug. There were higher incidences of gallstones, however. I think at the end of the day, it's important to note that independent experts did review certain categories of safety events, and these events pertain to hepatic safety, cardiovascular safety, and renal safety. There was a numerical increase in the number of adjudicated hepatic safety events, but most were mild in severity and related to biochemical changes. There were no severe or fatal adjudicated hepatic events in any treatment arm. As far as kidneys go, frequency of adjudicated kidney events and adjudicated major cardiac adverse events were balanced across the treatment groups. In summary, I certainly didn't necessarily expect this drug to have a positive histopathologic impact on the primary endpoint, which was regression of fibrosis by one stage in this tough-to-treat population. I am a bit encouraged by the safety data, however. Potentially that has implications and read-through to the F2-3 population that they're seeking approval in. Let me just stop there and get thoughts from the group.
I'm going to jump in here because this one interested me, given our conversations that we have regularly in stabilisation of disease. And of course, this is the only trial that's been read out that actually is the cirrhotic population. There was some positive movement in FibroScan, I believe. And I think you're right. An F1 improvement in this population is really, really hard. So maybe we are still addressing the wrong endpoint in this population. But I'd like to certainly have more information about, did it stabilise stabilise? Did it improve some outcomes for that population? Did it reduce portal hypertension? Did it improve their quality of life? Because this is the population that's the biggest risk factor. And I think Akero, from understanding, do have a phase 2b study, Symmetry, which is in biopsy-proven F4. So apart from that, there's no other of the ones we've read out today that are addressing the cirrhotic population. So I think it's a big hurdle to jump. And I think stabilisation and benefits we need to be looking at, not just it didn't meet its primary endpoint in this case.
Let me be a little bit reviewer number 2 and tougher on this situation. Despite the FibroScan improvement, this is clearly a no-go drug for cirrhosis based on efficacy. Even if you have a FibroScan improvement, itching was up, I think, to high 50s, albeit what was mentioned in the selection of people stopping. But still, it's a lot of itching for me for a cirrhotic patient. I don't recall if they had also more gallbladder side effects if they were on the drug or not, and it did not meet primary efficacy, which was expected. Even their safety data compared to F2, F3 patients, sure, it might help them, but at this point, with the amount of other positive results, I would advise them to go into combination therapy with this drug, even if they get approved, because the bar is much higher now.
Jörn, you have anything? I was going to make one really quick comment, and then we'll go to close. My quick comment was going to be that this is not significant. Stephen pointed out on the Axcella trial that there was a comparison between a group of 17 and 39% that was also not statistically significant. That's entirely, for those who are not cognizant of statistics, that's entirely a power calculation based on sample. The difference between 9 and 11 not only isn't statistically significant, but it's not in any sense meaningful. 17 and 39 is meaningful. You just have to power up to prove it's right. I agree with Stephen and Mazen. Louise, we'll see, but I'm pretty confident there'll be better options pretty quickly.
I think my final thought here is this is the largest cirrhosis study we've seen. It enrolled over 900 patients. That exceeds the Stella 4 study by a little bit. It's really been a tremendous effort. I mean, the important message is those patients are out there. I agree with Mazen that this is probably not the drug that's right for them, but for sure we have to address them at one point, and it remains to be a challenge to regress cirrhosis in a compensated patient. It's a big trial, and I think we'll learn more of it beyond the negative results.
And they wrote in the press release that they plan to continue analysis to figure out what else they can glean from the patient population.
Well, I mean, one thing we didn't hit on was, did they prevent progression of disease, which is very important. That's just more data that needs to be analyzed, but more to come there. Maybe they have a positive impact in that regard.
Yeah, I was going to say, in the category true confession, I don't think that there's a close on this conversation. I think the conversation is the close. We each of us have said, wow, at least once during the course of it, and it's going to just create an amazingly rich AASLD, and I think Stephen's right. It's been a banner month for NASH. That'd be my close. Anybody have anything they want to add?
Nothing to add. Thanks for running us through this, Stephen. It's been great as usual.
Yeah, thank you.
Yeah, Stephen, that was fantastic. Thank you.
It's been great being here, Roger. Thanks for having me.
Thanks.
All right, guys. Take care.
And we'll see everybody next week.
Take care. Bye-bye.
Bye.
And now for the Season 3, Episode 48 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. As this war takes more and increasingly odd twists and turns, let's hope it all comes out well for those who deserve for it to do so.
Thank you, Bulgaria and Slovakia.
This week, we can add 2 more countries to the list that have taken this podcast to heart. Last week, podstatus.com reported that Surfing the NASH Tsunami was the #1 medical podcast in Bulgaria, the #3 in Slovakia, and made the top 250 of all podcasts in all categories in both countries. Adding to Kuwait, North Macedonia, and Iceland on the list of where we've been #1 in several other countries. I don't keep that list, although I probably should, where we've cracked that top 250 overall. Dziękuję mnohokrát, Slovakia. Nagubodu Dorya, Bulgaria.
Episode 4 of our Rising Tide series comes out tomorrow.
This is an interesting, maybe even fascinating story. Last April, Mike Patel, president of the Canadian patient advocate group Fatty Liver Alliance and a spectacular advocate and educator for NASH patients, set out to have his own liver evaluated for NAFLD and NASH in a video series he presented on Facebook. His story and its twists and turns build an excellent case for the recent spate of guidelines and clinical care pathways more compellingly than simple paper can do. On the 4th installment of our Rising Tide and Diabetes series, Mike tells his story with his twists, turns, and confusion from different tests. And Ken Kusi and Jörn Schattenberg, 2 good friends of Surfing NASH who are intimately involved with guideline and pathway construction and education, comment. If you want to understand why it makes sense to start with FIB4 and then go on to one more simple fibrosis test, this is definitely worth a listen. Go to the surfingnash.com website to access it. Just a reminder, the NASH Tsunami and Diabetes Getting Ahead of the Rising Tide series is supported with a non-restricted educational grant from Novo Nordisk.
Our 100K party will record on October 31st and drop November 2nd.
As I speak, our Buzzsprout download count sits at 97,274, which means that sometime around mid-October, we will surpass 100,000 Buzzsprout downloads. I don't have a specific date, and we record our AASLD preview episode on October 24th, so we're reserving October 31st for our 100K celebration. On that podcast, we'll review 2.5 years of Surfing NASH history with some of our past hosts and guests. If you sponsored the podcast in the past or plan to do so, check your email. We'll have details and opportunities coming out shortly.
This week's Vault episode looks back at our NASH tag wrap-up on combination therapies.
This week, we're replaying Season 3, Episode 6, which was part of our NASH Tag wrap-up from January. In this one, I organized segments from 2 of the NASH Tag episodes to look at combination therapy for different windows: logical drug combinations, longitudinal drug combinations, and the use of combination biomarkers to diagnose and stage patients. Today's episode plays off some of those combination drug concepts that came up in the conversation. Enjoy it. And with that, I'm off. Thanks to our crew, Jake, Magic Mike, Eric, Steve, for pushing this episode and Rising Tide 4 out on a week where I'll be spending one day immersed completely in religious activities. These folks do a great job of getting our episodes out with increasing independence. And, as always, thanks to all of you, the listeners who make this such a worthwhile labor to pursue. We'll be back next week to talk about the recent NICE meeting on using FibroScan in primary care setting and what that tells us about treatment around the world. Until then, stay safe, surf on, and we'll see you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.